Intracellular transduction is the process where information outside the cell is transferred into the cell. When a cell acts upon stimuli from outside the cell using receptors, it is known as a cellular response. Ligands help with activation of signal transduction by binding to a central metal atom to form a coordination complex. The info outside the cell is conveyed into the cell through many messenger systems. Step 1 is the resting state. The receptor is not bound to the ligand, and the G subunit or gamma subunit is bound to the GDP. One system uses hormones that bind to the surface of the receptor. The second messenger system, known as a metabotropic messenger system, is cyclic AMP, which uses a hormone receptor, adenylate cyclase, and a G protein. In step 2, the ligand binds to the receptor, which then binds to a G protein. The gamma subunit releases GDP and gains a GTP. In step 3, the gamma subunits separate. Heterotrimeric G proteins are membrane proteins that bind to guanosine diphosphate, GDP, in its inactive state. The alpha subunit bounds to the inactive G protein so that it can be released from the receptor. These G proteins are required for the activation of adenylyl cyclase. In step 4, the G protein subunit activates or inhibits the target protein, which starts the signal transduction process. Cyclic AMP is produced from ATP, which results in the activation of protein kinase A, a type of enzyme. ATP is made during phosphorylation when a second phosphate is added. Phosphodiesterase is when an enzyme breaks the bond of a phosphate group. In step 5, the gamma subunit hydrolyzes its bound GTP to GDP and becomes inactive. A beta subunit undergoes a sequence of protein conformational changes that lead to the formation of ATP from ADP. In step 6, the subunits recombine to form an inactive G protein.